Hello and welcome. Today, we are diving deep into what I think is the most important molecule in the entire universe, DNA. It's literally the blueprint that makes you, well, you.
So let's start with a really big question. What is the instruction manual for every living thing? I mean, from a tiny, tiny bacterium to a gigantic blue whale, there has to be some kind of master code, right? And that code, the one that carries all of our hereditary information, is a molecule called deoxyribonucleic acid, or as we all know it, DNA.
To really understand this incredible blueprint, we've got to start with its alphabet. So, let's jump right into our first section, DNA's building blocks.
You know, every massive structure starts with a single brick. And for DNA, that brick is a little molecule called a nucleotide. This is the fundamental unit, the basic piece that links together over and over and over again to build the immense chains of our genetic code.
Okay, so each of these nucleotides has three distinct parts. First up, there's a 5-carbon sugar called deoxyribose. Hey, that's the D in DNA. Then you've got a phosphate group, which is kind of like the glue that links everything together. And finally, the most important part for storing information, a nitrogenous base. Now just imagine thousands, even millions of these linking up in one long chain.
That nitrogenous base, that's where all the action is. It's the letter in our genetic alphabet. And what's absolutely incredible is that this entire complex alphabet only has 4 letters. Adenine, which we call A, guanine, or G, cytosine, C, and thymine, T. Every single instruction for building and running an organism is written with just these 4 molecules. Amazing, right?
Now here's a really critical detail you're going to want to remember. These 4 letters aren't all the same size. They fall into 2 chemical families. You have adenine and guanine, those are called purines, and they're the bigger ones with a double ring structure. Then you've got cytosine and thymine, the pyrimidines, which are smaller, with just a single ring. Keep this size difference in your back pocket. It turns out to be a massive clue for the puzzle we're about to solve.
So, how in the world did we figure all this stuff out? Well, it was actually this fantastic detective story. So let's get into section 2, a scientific puzzle. By the middle of the 20th century, scientists knew all these little pieces existed, but they were locked in this really fierce race to figure out how they all fit together.
This timeline really sets the stage, doesn't it? I mean, DNA was first found way back in 1869, but for decades, nobody really got how important it was. Get this, even by the 1940s, scientists were still arguing about whether our genes were made of protein or DNA. By the early 50s, the race to find its structure was on, and finally, in 1953, the puzzle was solved.
The first huge clue came from a biochemist named Erwin Chargaff. He found something so strange, but so consistent, that his discovery became known simply as Chargaff's rules.
So, Chargaff analyzed DNA from all sorts of different species, and he kept finding this incredibly consistent pattern. It didn't matter what he was looking at, the amount of adenine always, and I mean always, equaled the amount of thymine, and the amount of guanine always equaled cytosine. A always matched T, G always matched C. No one knew why, but it was a solid, undeniable rule.
And here it is, the second game-changing clue. This is the famous photograph 51, an X-ray image of DNA taken by the brilliant crystallographer Rosalind Franklin. Now to you and me, it might just look like a blurry X, but to a scientist at the time, that X was a dead giveaway. It basically screamed that the DNA molecule had to be a helix, like a spiral staircase or a corkscrew.
Okay, so all the clues were on the table. We had the nucleotide building blocks, we had Chargaff's A equals T and G equals C rule, and we had Franklin's X-ray image showing a helix. This brings us to the big breakthrough moment in section 3, the double helix.
And this is it, the solution. In 1953, 2 scientists, James Watson and Francis Crick, proposed this model. It was a double helix, and its structure was just so elegant because it explained everything. It fit Chargaff's rules perfectly, it matched Franklin's X-ray photo, and it made complete chemical sense.
So, how does it all work? Just imagine a twisted ladder. The 2 long rails of the ladder, those are made of the sugar and phosphate parts. And the rungs in the middle, the steps you'd climb, those are the bases, the letters. The 2 strands run in opposite directions, kind of like 2 lanes on a highway. And here is the absolute genius part. Adenine, the big one, always pairs with thymine, the small one. And guanine, the other big one, always pairs with cytosine, the small one.
This perfect pairing creates rungs that are all the exact same width, which just so happens to perfectly explain Chargaff's rules. See how it all just clicks?
It's a beautiful structure for sure, but in biology, form always follows function. The elegant shape of DNA is the absolute key to everything it does. So let's explore why in Section 4, Structure as Function.
It also turns out the double helix isn't totally static. It's a dynamic molecule. While this B form you see in the middle is the standard version we have in our cells, DNA can actually twist itself into other shapes, like the more compact A form, or even this weird left-handed Z form, all depending on its environment.
Okay, get ready for a mind-blowing fact. If you took the DNA from just one of your cells, one single cell, and stretched it all the way out, it would be about 2 meters long. So how on earth do you fit a 2-meter string inside a microscopic nucleus? The answer is supercoiling. The cell just twists and coils the DNA on top of itself, packing it into an unbelievably tiny space.
But the most profound, the most important function of the double helix is its ability to copy itself. It's just perfectly designed for it. The two strands simply unwind and separate, like a zipper. Then, since A can only pair with T and G can only pair with C, each of those original strands acts as a perfect template to build a new partner. And what's the result? 2 new, absolutely identical DNA molecules. This right here is the very basis of life and heredity itself.
So, why does all this molecular detail really matter to us? How does it affect our daily lives? Let's bring it all together in our final section, clinical significance.
Well, because DNA is the master blueprint, any error in that code can have really devastating effects. A change in just a single letter, what we call a mutation, can cause genetic disorders like sickle cell anemia. And over a lifetime, the accumulation of damage to our DNA is a primary driver of diseases like cancer. It's that fundamental.
The discovery of the double helix was like learning to read the book of life. It showed us how information is stored and passed down through generations. It is, without a doubt, the code of our past, written into every single cell of our bodies. But now, with our growing ability to not just read that code, but also to edit it, well, that leaves us with a pretty profound question for the future, doesn't it? What will we choose to write?